So hello again. We'll go ahead and get started. Welcome back to those of you who have participated in last week's session, and welcome to those of you who are new for this week's session. This is Ellen Webb with the Center for Environmental Health. On behalf of CEH and Reproductive Health Technologies Project, we welcome you all to the second session in the 6-week webinar series. As I mentioned last week, we will be hearing from leading national experts in reproductive and developmental health. And in particular, we will be focusing on resource extraction and energy production, discussing potential adverse reproductive health and developmental health effects in humans, especially as it relates to maternal and fetal development. For today's session, we will be discussing research that is being done exploring the link between unconventional oil and natural gas operations and developmental and reproductive health. We have some great participants today, and in a moment, I'm going to be turning this over to Carolyn Cox, our research director at CEH, who will be moderating today's session. But before I do, I would like to introduce Carolyn. Carolyn leads CEH's research on toxic exposures, identifying, analyzing, and substantiating the scientific bases for our work to eliminate threats to children and others exposed to dangerous chemicals and consumer products. Under Carolyn's leadership, CEH has won dozens of landmark legal agreements with major companies, requiring them to eliminate harmful chemicals from their products and/or reduce their toxic emissions. Carolyn has testified to Congress and to state and federal regulatory agencies on consumer product safety and health risks from pesticides. She has co-authored science articles in peer-reviewed journals on hidden ingredients in pesticides, air quality around fracking sites, and on the successes of CEH's work. Previously, Carolyn worked for 16 years as a staff scientist at the Northwest Coalition for Alternatives to Pesticides in Oregon. She was also editor of Northwest Coalition for Alternatives to Pesticides Journal of Pesticide Reform. Carolyn represents CEH on the steering committee of Californians for Pesticide Reform. She also serves on the board of directors of Beyond Pesticides. She writes and speaks regularly as a national expert on the toxicity of and alternatives to pesticides. Carolyn has a master's degree in entomology from Oregon State University and is a graduate of Swarthmore College in Pennsylvania. So, Carolyn, welcome. I'm going to turn this over to you in just a moment so we can now begin.
Hi, everybody. Welcome to the webinar. I'd like to introduce our first speaker, who is Dr. Susan Nagel. She is an associate professor of obstetrics, gynecology, and women's health at the University of Missouri School of Medicine. Her previous education is a biology degree from Hendrix College and PhD in physiology from the University of Missouri, as well as postdoc from Duke University in North Carolina. So her research has focused on the developmental origins of adult disease, meaning what happens to you as before you're born and as a young child can cause problems later in life. And she also works on endocrine disruption, meaning how chemicals can disrupt our hormone systems and with respect to hydraulic fracturing, she's published research on finding estrogen and androgen activities of hydraulic fracturing chemicals, and also in the surface and groundwater near places where drilling is going on. I think she was the first person to conduct this kind of research that showed an association between endocrine disruption activity in water and spills that were related to oil and natural gas processes. Right now, she's conducting research on hormonal activity in water near fracking sites, and also in the laboratory studying the reproductive toxicity after developmental exposure to a mixture of chemicals used in oil and gas production. So, welcome, Dr. Nagel.
Thank you very much to the organizers for the invitation to participate today. I am, let's see, can you see my screen yet? Great.
There you go.
Thank you. All right, thanks. Sorry about that. Excellent. So as we heard, I'm going to speak to you today about endocrine disrupting activity associated with unconventional oil and natural gas operations. So research in my lab has focused for about 20 years on endocrine disrupting chemicals, and I have taken a long range view of developmental origins of adult disease, and only in the last 5 years have I brought into the mix oil and gas extraction processes that may contribute chemicals that are endocrine disruptors. So what is an endocrine disruptor? The Endocrine Society defines it as a chemical or mixture of chemicals that interferes with any aspect of hormone action. And this is slightly different than the EPA definition. However, it's the one that I'll use today. So it's a chemical that has the potential to disrupt any aspect of hormone action. So EDCs are found in many, many products. They're naturally occurring. They can be heavy metals. They can be plant-produced chemicals, they can be industrial chemicals, and of course they can be designed to disrupt the endocrine system like pharmaceuticals. Pesticides and herbicides are a large group. So one thing, or two major ways that endocrine disrupting chemicals work are to either bind directly to hormone receptors, and shown here are the estrogen and androgen receptor, which are what we focused on to the normal response of a hormone. They can also alter the amount of the hormone in the body, so they can increase or decrease the level of hormones in the body. So these are the major ways that we think of that endocrine disruptors work, although there are other ways. So steroid receptors, and what do I mean by steroid receptors? They are estrogen receptors or androgen receptors, or we have quite a number of receptors that use the steroid receptors or nuclear receptor superfamily. And so what they do is they bind directly to regions of DNA, in this case an estrogen response element, and recruit the machinery that is needed to regulate gene transcription. So in short, they are involved in turning on or turning off genes in the right place at the right time. So hormones like estrogen, and shown here, estradiol is the principal physiological estrogen in people and many other animals. It's a very small lipophilic hormone. So because it likes to dissolve in lipids, it's mostly carried in blood by sex hormone binding globulin and albumin. And it's only this small free fraction that is able to passively diffuse out of the bloodstream and into cells and bind to those intercellular estrogen receptors. And a common stimulation or common response is stimulation of cell growth or cell division. So we can see here increased cell number in the endometrium as a response of estrogen action. So why is endocrine disruption important to human health and disease? So there are three main reasons that I want to touch on. So hormones and endocrine disrupting chemicals can act at very low concentrations. So here's estradiol, which I just mentioned is the principal physiological estrogen that is working in the part per trillion range. So chemicals, like endocrine disrupting chemicals, and shown here is bisphenol A, which is a poster endocrine disruptor child, if you will, can work in the part per billion range. So while often endocrine disrupting chemicals are weaker, it takes more of them to stimulate the same response. we are often exposed to higher concentrations of these chemicals. So for example, most of us have higher concentrations of bisphenol A in our blood than estradiol. So the second region is that human exposure can be within this range of bioactivity. And finally, developmental exposure can alter adult health and disease. So hormones are essential for normal health and development. It is their proper timing and the proper amount of exposure that regulates how we develop. So it is the presence of fetal testosterone that resulted in sexual differentiation of Brad, and the absence of fetal testosterone that resulted in sexual differentiation of Angelina. So while those are very extreme examples of developmental programming, nevertheless, I think they illustrate the point very well that hormones are essential for normal development. Fetal and early life exposures to EDCs is associated with adult disease. So many diseases have been linked to fetal exposures. So while we focus mostly on pollutants, we know a huge amount about nutrition, undernutrition, overnutrition, improper nutrition during development, and also maternal health and disease, and how all of these factors can influence the fetus and result in increased susceptibility to adult diseases. So all of this led me to ask the question about unconventional oil and natural gas extraction as a potential source of endocrine disrupting chemicals. So I think that most of you know, but just to get on the same page of what we're talking about today. So unconventional oil and gas extraction is really the combination of hydraulic fracturing and horizontal drilling. So drilling down thousands of feet underground, turning and proceeding either horizontally or at an angle to the surface. This allows drilling through shale and coal bed layers that contain natural gas that was previously inaccessible or unprofitable to extract. By pumping down millions of gallons of water under high pressure, we can fracture the underground rock to release the trapped natural gas. And this process is extremely effective. Now, in order to do this, hundreds of thousands of gallons of chemicals are added to the mixture for a variety of reasons. So while the majority of the constituents are water, millions of gallons of water, we have thousands of gallons of chemicals that are added to this mix and proppants to hold open those fractures. So I became interested in this topic when Colburn and her colleagues first drew our attention to the potential for these chemicals to be endocrine disruptors. They cataloged the published literature on potential effects of chemicals used in the process. So when they started this in the late 2009, 2010, they looked at 350 chemicals where the chemicals were known. So they had an actual chemical identification number. Otherwise, it's almost impossible to look for effects of these chemicals in the literature. So of those 350 chemicals, you can see that many of them, over 80%, had some indication that they could interfere with the respiratory system, skin, eye, sensory, and so on. For endocrine disrupting chemicals, over 35% of this 350 chemicals had some evidence in the literature that they could be endocrine disruptors. So the combination of drilling and under high pressure results in millions of gallons of wastewater. So the flowback is the water that returns immediately to the surface, and produced water returns over the life of the well and contains fracking chemicals, liberated salts, and radioisotopes from deep underground. So this leads to the potential contamination of surface and groundwater. So wastewater spills and leaks are common. There is often direct disposal, as shown here, of wastewater into surface water. whale casings can fail, and there also can be underground migration of fluids. This led us to our hypothesis that surface and groundwater at natural gas drilling spill sites might contain more endocrine disrupting activity than reference sites. So this is a map of Garfield County, Colorado, and the box shows our first study that we did in 2010 and the sites within it. The red dots are active natural gas wells, and our sites are highlighted here in white letters. We also had reference sites outside of this drilling dense area and upstream in the Colorado River. So for our 2010 sample collection sites, we targeted very specifically sites that had some sort of incidents, some sort of intentional or unintentional release of this wastewater. And you can see that these were over time. So these were not all in 2010. Only one of them had actually happened the year and a couple of months before we were there. So what we do is we collect surface and or groundwater at each site. We collect it in amber glass bottles. We bring it back. We keep it on ice. We bring it back to the lab, and we do what is called a solid phase extraction to concentrate the chemicals present in the water down to a small volume. And in this case, we're using them in a cell-based reporter gene assay. And so what that means is that the more endocrine disrupting activity, ultimately, we can use the fact that estrogen receptors or androgen receptors turn on or off gene transcription. And in this case, we engineered them to turn on or off a gene that can produce light. So in our first study, we measured groundwater activity both in a Missouri reference site. So this was actually at a USGS well here outside of Columbia, Missouri. We measured a Colorado reference site. So this was in Garfield County, but outside of this area without any known spills. And we measured 3 sites that had known spills. And so in this particular case, we saw increased estrogenic activity in groundwater and increased anti-androgenic activity. So that means that that activity is opposing the normal action of androgens, or male sex hormones. In surface water activity, we saw a slightly different trend. So we saw some estrogenic activity, but we saw a huge increase in anti-estrogenic activity, and once again, the presence of anti-androgenic activity, which we never saw in our reference sites. So with this, we had found an association between spill sites and endocrine disrupting activity in surface and groundwater. We published this last year. Chris Casotis, a senior graduate student in the lab, was the first author. What we did next was go back to Garfield County in 2014. Our goal here is a much larger, more comprehensive sampling effort. So we measured 48 different sites in Garfield County. And this analysis is currently underway and will have results hopefully by the end of the year. Another collaboration that we are involved with is looking at endocrine disrupting activity in surface water near a disposal facility in West Virginia. And in this particular case, the design is such that a creek flows through an injection well pad. So upstream of this injection well pad is a background site. And this creek flows through this site. You can see there's some sample retention ponds here, and the injection well is actually right here. And this site has quite a history of disposal of oil and gas extraction fluid. So we measured endocrine disrupting activity in each of these locations marked with numbers. So what we can see, first, let me orient you to this graph. So now on this particular graph, I'm showing you 3 types of activity: anti-thyroid hormone, anti-progestogenic hormone, and anti-estrogenic activity. First, the blank. So this is just what's naturally in water and in our tubing and in all the components of our assay. So you can see that we had a little bit of background activity in our blank. but very little. Next, at site 4 was our background field site location, and we had very, very low endocrine disrupting activity at that site upstream of this injection well. So next, at site 6 on the pad, we start seeing increased activity of anti-progestogenic and anti-estrogenic activity. However, once we get slightly downstream of where the collection ponds and storage and evaporation ponds had been, we see a dramatically elevated level of endocrine disrupting activity. This water at this site was tenfold more diluted than in Colorado. So in this case, where we see actual disposal of these wastewaters and evaporation, these have resulted in a dramatic increase in endocrine disrupting activity. They are also commensurate with dissolved methane in the water, suggesting their source from waste. So our hypothesis, we have expanded. Chemicals used in hydraulic fracturing will disrupt hormone receptors for estrogen, androgen, glucocorticoid, progesterone, and thyroid hormone. So Chris Cassotis, what he did next was to test 24 chemicals that are known to be used in hydraulic fracturing and other natural gas processes. So he tested these 24 chemicals for those 5 nuclear receptor activities, and he measured the ability to activate them or to inhibit them. And shown here is a partial list of the chemicals that he used. So what I'm going to show you is a series of the 5 different receptor activities, and not so much as what I won't focus on is individual chemicals so much as the overall ability of these chemicals to disrupt the endocrine system. So solid bars here are chemicals that are at a concentration tenfold higher than the hatched bars. So it showed some sort of toxicity in our cell-based assay. So these hatched bars actually are at a concentration tenfold lower than the solid bars, showing that they have even greater activity to disrupt, in this case, the estrogen receptor. And what you can see is that most of these chemicals have some ability to inhibit normal estrogen receptor activity. You can shift your attention to this 23 mix. So that is a mixture of the 23 chemicals so in this we had tested 24 chemicals, and so 23 of those chemicals here showed maximal inhibition of the estrogen receptor. And what you'll notice is this says 24 mix. And so that's because the 24 mix actually has bisphenol A, which is a very positive stimulator of the estrogen receptor. So it is actually inhibiting some of this estrogen receptor inhibition. Next, looking at the androgen receptor. So this is the ability of these chemicals to inhibit the androgen receptor. So what you may not know about bisphenol A is that it is a very good inhibitor of the androgen receptor. So in this case, many of these chemicals also can inhibit the androgen receptor. Many chemicals can inhibit the progesterone receptor, although fewer, and fewer still can inhibit the glucocorticoid receptor and the thyroid receptor, although still significant number. So here's a summary of what we saw. So one chemical could stimulate the estrogen receptor, and this was bisphenol A, 1 and 2 for the progesterone and thyroid hormone receptors. However, many chemicals could inhibit those receptors. Shown here is the combination of those 23 chemicals. So now we're not looking at one chemical and its ability to inhibit the receptor, but we're looking at a mixture of 23 chemicals. So all 23 of those chemicals minus bisphenol A. And so what we can see here is that this mixture had the highest level of activity for inhibiting the estrogen receptor. However, all of the mixtures could inhibit every receptor that we tested. Now what Chris wanted to do then was ask the question, could this mixture alter development in our mouse model system? So what he did was to use this mixture in a developmental study. So shown here is, this is a timeline showing prenatal life. So this is looking at conception, and then on gestation day 11, we put chemicals in their water and the dams, the mother's drinking water, and then shown here is the day of birth, which is about, mice have about a 19-day gestation. So here is our mixture of chemicals. What we did then was put each of those chemicals at the concentration shown here at 4 different levels. Then in adulthood, we examine different parameters. And what I'm going to show you is, first is looking at the ability of these chemicals to inhibit sperm production. So this is looking at the caudal epididymis isolated at the time of necropsy, and the sperm were allowed to swim out and they were counted. So what we can see is that developmental exposure of these males in decreased sperm counts in adulthood. So looking at mixed forward, this was our lowest level of exposure. And what Chris has estimated is that from the very little that has been published on what is the concentration of these chemicals in drinking water, what we see is that mix 2 and mix 3 are probably within the range of some known groundwater contamination sites of some of these chemicals, like for example benzene. He also saw elevated testes weight both at postnatal day 21 and in adulthood at postnatal day 85. And this study is ongoing, and we are looking at the female offspring as well. We have also initiated a whole series of additional studies to look at female reproduction. With this same mixture. To do is identify chemicals from our field study that are in the water at those sites, measure their endocrine disrupting activity, and then use those in the lab. And so one of the ways that this is, we talk about this, is an effects-directed analysis. So we have, we can test our samples from the field, and we can use these to identify the ones that have the highest endocrine disrupting activity. And then we can identify unknown chemicals in these fractions that have the highest level of activity and test them in our bioassay to determine which ones are most likely to be responsible for that endocrine disrupting activity. Preliminary results of this approach are shown here. So these are the chemicals that I initially showed you in our mixture. So we have the chemicals highlighted in yellow. We have identified in wastewater samples, for example, from Colorado, where we have collected wastewater and measured very specifically these chemicals. So in conclusion, the hormones and EDCs can work at very low concentrations. So EDCs are associated with disease in people, and of 24 fracking chemicals tested, 23 were endocrine disrupting chemicals. All 5 hormone receptors that we tested could be disrupted by chemicals used in oil and natural gas processes. And surface and groundwater from hydraulic fracturing preventable discharge sites had greater endocrine disrupting activity than control sites. I'd like to thank a few I mentioned, and the rest are shown here. So with that, I am going to say thank you and turn it back over to our moderator.
Thank you so much, Susan. That was wonderful. Really fascinating subject. I'm so glad you're doing that research. Our next speaker is Dr. Lisa McKenzie. She is an assistant research professor at the Colorado School of Public Health. Her previous education, she has a BA in chemistry from the University of Colorado, a master's of public health from the Colorado School of Public Health, and a PhD in environmental chemistry from the University of Montana. She teaches now classes in risk assessment and environmental health. Her research has looked at how air pollutants and other exposures come from unconventional oil and gas development could affect health people living in areas that oil is occurring. She has a cohort study investigating the relation between what called birth outcomes and how close those moms lived to natural gas development. Is one of the first of that type of study published, and she found that there is the potential for a variety of health outcomes, including related to breathing, related to nervous system function, and related to development that result from exposure to air pollution caused by natural gas development. So, welcome, Lisa.
Thank you, Carolyn, for that introduction, and thank you, Ellen, for the invitation to share my results today. Okay, thank you. So what I'm going to present today are the results of our study on congenital heart defects in infants born to mothers living in proximity to natural gas development in Colorado. And then I also am going to talk to you about our plans for the next steps in this study, so what we're going to be doing next. So if born in Colorado, at least 2 of the 135 infants shown here would likely have a congenital heart defect. The Colorado prevalence of congenital heart defects is 18 per every 1,000 births, which is about twice the national prevalence rate. Congenital heart defects are the leading cause of death due to birth defects, and the healthcare costs are high at about $1.4 billion each year. The cause of this most common type of birth defect is not well understood. They are thought to originate in the first 2 months of pregnancy as a result of an inherited genetic trait or an interaction between genes and environmental stressors. Many environmental stressors have been implicated, such as maternal smoking and maternal exposure to solvents. Exposures to hazardous chemicals also have been implicated. Maternal exposures to hazardous chemicals may affect the developing fetus, particularly in the first 2 months of pregnancy. Several studies have associated maternal exposure to toluene, xylene, and benzene with increases in the prevalence of birth defects. Other studies have associated maternal exposures to air pollution with some types of congenital heart defects. Oil and gas development is an industrial process resulting in community exposure to multiple chemicals and other environmental stressors, some of which have been associated with birth defects. Diesel engines power heavy equipment used for worksite development, as well as transport large volumes of water, sand, and chemicals to sites and for waste removal. Studies in Colorado, Texas, Wyoming, Ohio, and Oklahoma have demonstrated that oil and gas development compounds, nitrogen oxides, sulfur dioxide, particulate matter, and polycyclic aromatic hydrocarbons, and these occur from either the well itself or from associated drilling processes or related infrastructure. So we're talking related infrastructure would include drilling muds, hydraulic fracturing fluids, tanks that contain wastewater and liquid hydrocarbons, diesel engines, compressor stations, dehydrators, and pipelines. Some of these pollutants, for example, toluene, xylenes, and benzene, are suspected to be teratogens or mutagens, and they're known to cross the placenta. This raises the possibility of fetal exposure to these and other pollutants resulting from oil and gas development. The development and production of oil and gas has occurred very near homes. Until recently, Colorado regulations allowed wells to be located as close as 150 feet from a home. And to put this into perspective, this is about half of a football field. With the new regulation, 500 feet. And this map shows the extent of oil and gas development in Colorado. And you saw in Dr. Nagel's slide, Garfield County in the northwest of the state. The red here are active wells, and the green indicate permitted wells. And historically, most of the development has been located in the rural areas of Weld County in the northeast and Garfield County in the northwest. However, it's becoming increasingly common for natural gas development, oil and gas development, I should say, to encroach onto populated areas, and that's shown here in this slide. The blue circle denotes the location where there is now a multi-well pad, and you can see the neighborhoods around this site. This potentially exposes more people to air and water emissions, as well as noise and community level changes that may arise from this industrialization. The growing potential for maternal exposure to air pollutants from oil and gas development and the vulnerability of the developing fetus to environmental stressors motivated us to conduct our first study. Colorado's systematic tracking of data on both birth defects and oil and gas development also was a motivating factor. Our goal was to explore the association between maternal exposure to natural gas development and congenital heart defects using a data set with individual level birth data and geocoded natural gas well locations. Using a retrospective cohort study design, we determined past individual maternal exposures to natural gas development as proximity to a natural gas well. We then followed each mother to determine whether or not there was a congenital heart defect in the birth of her infant. Our cohort included almost 125,000 infants that were born between 1996 and 2009. The mothers of these infants lived in rural areas of Colorado. The mothers living in metropolitan areas with populations of 50,000 or more were excluded, so this excluded the Denver metropolitan area. The cohort was restricted to white Hispanic and non-Hispanic mothers because over 95% of the mothers fell into these two ethnic categories. The cohort also was restricted to singleton live births. And now I want to explain to you how we defined exposure. We defined exposure as the existence of one or more gas wells within 10 miles of the mother's residence. So that's designated by the two circles here on the graph. And so we then used geocoded well locations available from the oil and gas information system, and we identified all the existing wells that were in 10 miles of the mother's home. The mother was considered to be unexposed if there were no wells within this 10-mile radius shown on the right-hand side of the screen here. By existing well, we meant that there was evidence that the well was present and had not been abandoned in the year the infant was born. And this is very close to the Colorado Oil and Gas Conservation Commission's definition for an active well. Distribution of the wells within the 10-mile radius of the maternal residence showed that 50% and 90% of wells were within 2.3 and 7.7 miles of the mother's home, respectively. We then used an inverse distance weighted approach, commonly used to estimate individual air pollutant exposures from multi-fixed locations to estimate what the mother was exposed to. In the next few slides, I will walk you through how we calculated the inverse distance weighted well count. As shown here, we began by measuring the distance of each well from the mother's home. Then we calculated the inverse of the distance by dividing 1 by the distance of the well from the mother's home. We did this for each well. Then we counted up all the inverse distance weights for all the wells within the 10 miles of the mother's home. Inverse distance weighting gives more weight in the count to those wells that are closest to the home. So the wells closest to the home have more influence in the exposure measurement than those that are further away from the home. Here are some examples for 4 wells within 10 miles of the mother's home. If each well was 1 mile from the mother's home, the inverse distance weighted count would be 4. If all the wells were 5 miles away, the count of the inverse distance weights would be 0.8. And if 2 wells were 1 mile away and 2 wells were 2 miles away, the inverse distance weighted count would be 2.4. So you can see that using the count of the inverse distance weights, exposure measure each of congenital heart defects in each of the 3 exposure groups, the low, medium, and high group. The analyses were adjusted for potential whether or not the mother smoked or used alcohol. The elevation calculated an odds ratio. An odds ratio greater than 1 indicates that there's a positive association between the exposure and the congenital heart defect. An odds ratio less than 1 indicates that there's a negative association between the exposure and congenital heart defects. And finally, an odds ratio of 1 indicates that there is no association. We also considered the trend in the odds ratios from low to high exposure. So this is our first result, and as shown here by the pie chart, the births were approximately evenly divided between exposed and the unexposed group. 47% of births in rural Colorado between 1996 and 2009 were to mothers with 1 or more existing wells within 10 miles of her home. This result confirms that a large proportion of Colorado's rural population resides in areas with oil and gas. And so now I'm going to move on to the results of the logistic regression and the odds ratios that we calculated. And the odds ratios are shown here on the y-axis, and an odds ratio of 1 at which you would expect no association is shown on the horizontal axis. Our adjusted estimates indicate that there's a monotonic increase in the prevalence of congenital heart defects with increasing exposure to natural gas development or increasing maternal proximity to natural gas development. Births to mothers in the most exposed tertile, shown here, had a 30% greater prevalence of congenital heart defects than to mothers with no wells within a 10-mile radius of their residence. We saw an even stronger associations when we did an exploratory analysis of specific heart defects by clinical diagnostic groups, and these are shown here. These exploratory analyses showed an increased prevalence of pulmonary artery and valve defects, is shown up here on the right, by 60%. Ventricular septal defects by 50%, but valve defects very low numbers. While our preliminary study suggested an association between congenital heart defects and maternal proximity to oil and gas development, it did have limitations that may have influenced the results. Therefore, we are conducting a second study to address these limitations. In our next study, we are developing rigorous exposure assessment approaches that will link specific well activity and emission models to birth data to estimate maternal exposures in the first 3 months before conception and in the first 2 months of pregnancy. One of the limitations of the first study was that we had grouped all congenital heart defects together. This may have made the difference between exposed and non-exposed groups harder to see. In the current study, we plan to divide the congenital heart defects into diagnostic groups and conduct a case control study for each diagnostic group. That is, we will conduct 5 case control studies. The case control study design is helpful in investigating rare conditions like congenital heart defects. Even though congenital heart defects are the most common type of birth defect, they are still a rare condition. And then instead of having to investigate all 175,000 births in our cohort that we have now, we can focus on a relatively small number of births, approximately 1,500 births, which will allow us to fully describe the mother's exposure for both the cases and the controls. The cases will be infants with congenital heart defects, and then the controls will be infants that don't have congenital heart defects, and they will be matched to the cases. Another limitation of our initial study is that we evaluated the density of. However, the critical time period for the formation of congenital heart defects is right before pregnancy and in the first 2 months of the pregnancy. Therefore, in this study, we will evaluate maternal exposures to oil and gas development in this critical time period. Another limitation of all wells around the maternal residence to be the same because we did not know what was happening at the well. Because the case control study design allows us to reduce the number of mother-infant pairs, we will be able to evaluate Colorado and gas information system records to determine what was happening at each well around the mother's home in the first 2 months of her pregnancy. In addition, we can estimate the predominant wind direction, and we can also evaluate other air pollution sources that are within this 10-mile radius of the mother's home. With this information, we will assign an air toxics emission intensity to each well and to each other source of air pollution around the mother's home. Finally, we were also limited by lack of information on the mother's activities away from her residence and about what her occupation was. We didn't know much about the mother. So we will use a search whether the time she spent away from home was located near oil and gas development. So to conclude, it is important to continue the study of potential associations between fetal development and exposures from oil and gas operations. These operations are rapidly increasing in populated areas, and they're known to emit teratogens and mutagens. The human fetus is especially sensitive to environmental exposures. Our preliminary study indicates an association between maternal proximity to oil and gas development and congenital heart defects, which have significant health consequences. However, there were limitations in the preliminary study, and we plan to address those in our current study going forward. The co-investigators on the preliminary study were Dr. Roxana Witter, John Adgate, Lee Newman, and Ray Gao at the Colorado School of Public Health, as well as David Savage at Brown University. The preliminary study was funded by our department here, and the American Heart Association is funding the new study. Thank you for your attention.
Thank you so much, Lisa. Your research is just so important. I feel really privileged we got to hear about it. I wanted to invite everybody who's listening to the webinar at this point to ask questions of either Dr. Nagel or Dr. McKenzie. And the way you would do that is go to the chat box, which you should see on your screen, and just type in a question, and Ellen will be collecting those questions and then asking our panelists to respond. And while you're busy typing in your questions, I wanted to get things started by asking a few questions of each of our panelists. Susan, I thought I'd start with you first. I think you answered this briefly during your talk, but maybe you could give us just a little more detail. Why does endocrine disruption matter?
Sure, yeah. So endocrine disruption matters particularly for this topic because of the exquisite sensitivity of the developing fetus to hormones, and also adults. So even in adults, we have very low concentrations of endogenous hormones, such as estrogens. So when we have chemicals that are present at higher concentrations that may be weaker, they are present at concentrations that often can oppose normal estrogen action. And so for this topic, particularly where we have multiple chemicals that may be acting in the same way, we have a lot of evidence that those chemicals can act in an additive way. So they can add up and work on this very sensitive system.
Thank you. Lisa, a question for you, if you're willing. I know that the kind of study that you did can run into the frustrating situation, which happens a lot, where moms move. So they may not be living at the time their child was born in the same place that they lived while they were pregnant, or something like that. How much of a factor do you think that was in your study?
So other studies have looked at maternal mobility during pregnancy pregnancy, and they find that about 20 to 30% of mothers do move during their pregnancy, but most of those move within the same counties that they're living in. They're not moving very far away. And the studies that have looked at this have found that it has not made a large difference in the results. So based on those studies right now, I don't think it made a big difference, but I don't know that for sure. So one of the things we're going to be doing in this further study is one of the questions we're going to be asking the mothers on the surveys is about where they were living during the first 2 months of their pregnancy and just before their pregnancy. And we're going to look and see if there's a difference in results if we take that into account or not.
Great. Thank you. Ready to move questions from the audience, or should I keep asking questions?
Carolyn, I think we actually still have a little time, so you can continue to go through the questions if that's okay with the speakers.
Okay, great. Susan, would you be willing to give us an idea whether endocrine is more important than other health concerns that you might be looking at associated with oil and gas drilling?
Sure. So we know that chemicals used in the process and liberated by the process affect many, many health systems. So one of the things that we think about endocrine disruption is because of its sensitivity, or because of the health system's sensitivity to these chemicals, it's a very good sentinel, if you will, so canary in the coal mine almost. So with developmental exposure, with disruption, potentially of reproduction. These are really good endpoints because they're so sensitive to disruption. That's not to say that they are the most important or that they're more important than other endpoints. For example, cancer and other respiratory types of effects. However, they do have the advantage of being potentially the most sensitive, so they're good targets to go after as far as looking at potential health impacts of this process.
Great, thank you. Lisa, another issue that I'm sure came up in your study was, how do you define what an existing well is? And I realize that question might sound sort of naive, but I'm sure there must be issues with wells that were abandoned, maybe even abandoned, while a particular woman was pregnant, or I mean, lots of factors that make this more complicated than just a yes/no situation. And so how did you define an existing well, and is that different than how Colorado defines it?
So we defined an existing well as, is it a well that was physically present, so had been drilled and had not been abandoned. So we excluded abandoned wells from our analysis. The Colorado Oil and Gas Conservation Commission defines a well that is there and that has not been abandoned, and they include wells that are shut in meaning that they're not producing gas at the time. So the two definitions are very similar, if not identical to each other. And we made that definition for the year the child was born. So it is possible that a well could have been abandoned within that year of birth, but that's probably only would have been for a very small number of the wells that would actually happen. they're not abandoned at that high of a rate.
Thank you. See, I have one more question for Susan. Just, I mean, your research is a very compelling topic. Endocrine disruption has been, you know, an area of concern for the last couple of decades. But how exactly did you decide to pursue its connection with oil and gas development?
Really, it was the preliminary work done by the Endocrine Disruptor Exchange to highlight the huge number of chemicals used in the process and their huge potential to be endocrine disruptors. So they did such an amazing job synthesizing that literature that it was very compelling. And so with my basic interest in endocrine disruption, it was a natural fit and a natural question for us to ask.
This is probably just a good time to take a brief moment and just sort of acknowledge the Endocrine Disruptor Exchange and all of the great work that they've done, and you know, especially the founder, Theo Colburn, who you know, really made this her life's work and did an incredible job in making this the subject that it is today. And then, Lisa, I had one more question for you. Can you talk about confounding? And that's always an issue when you do your kind of epidemiological work. How would that affect your results?
So, as you said, Carolyn, confounding is always a limitation in epidemiological studies, and that involves having factors that either you weren't able to take into account that might be affecting both your exposure and the health measure that you're looking, or the health outcome that you're looking at. We did account for the information on other variables that was available in the birth record, such as maternal smoking and education. We didn't have any information, though, really on the socioeconomic status of the mothers, and so this could have resulted in some confounding of our results, and we're hoping to gather more information on that in the current study.
Great, thank you so much. Ellen, are you ready with some questions from the audience at this point?
Yes, I have a number of questions that have come in. So Dr. McKenzie, I have a question for you, and that question is, why do you include elevation of mother's home as an independent variable in your logistic regression model?
The reason why we included elevation of mothers' homes is in Colorado, most of the gas drilling occurs below 7,000 feet, and we have mothers in Colorado living well above that elevation. And elevation, when we did the first study, we looked at other effects other than congenital heart defects. We also looked at low birth weights, but elevation has been associated with low birth weights and some other birth outcomes, and that's the reason why we included elevation in our analysis.
Great. There's another question here, and this is actually directed to both panelists. And the question is, it seems to me that both your studies focused on maternal contribution to fetal development. Could you please let me know what you think about paternal contributions?
Sure.
Dr. Nagel.
So paternal contributions to the offspring are absolutely well documented. We have not examined those yet, although I think it's an excellent thing to do.
And I agree with that. Paternal exposures are important. We don't have as good of records of maternal exposures or databases on fathers. However, when I mention the survey, we will be asking about the father's occupation.
Great, thank you to you both. So I have a question here for Dr. Susan Nagel. The question is, many times we hear about brine water used in oil and gas production. It is described by industry as benign material. However, does this brine water contain endocrine disrupting material or other potentially known chemical contaminants? And have there been any specific studies on this?
I will first have to just say that if I have to guess, then it is wastewater. So what I mean by that is these things are, all the different kinds of wastewater, produced water, flowback are called many different things. However, it often contains very high levels of salts and are therefore called brine waters. So now to answer your question of have there been studies of what kinds of chemicals are there, absolutely. We have done a little bit of that research and are starting to analyze that. And a preliminary analysis of unknowns of some of that water looked at about 65 chemicals, many of those being endocrine disruptors. Others have published on this. Now, with all that said, there's actually not a lot of information, especially not a lot of published information on those waters, and certainly not on endocrine disrupting chemicals. However, we will be publishing a study here in the next couple of months looking at very specifically some produced waters and wastewater analyses of endocrine disrupting chemicals.
Great, thank you.
So yes, if those big flowback, yes.
Okay, great. Thank you for that. So we have one more question here for Dr. McKenzie, and the question is the prevalence of congenital heart defects in unexposed mothers in Colorado same as the general population in the rest of the United States?
In our study, the prevalence of congenital heart defects in the unexposed mothers in rural Colorado was higher than the national prevalence rate, but a little lower than the prevalence rate in Colorado. It was about 13 per every 1,000 births.
Great. Okay, thank you for that. So I think we're going to wrap up here. Thank you to everyone for your questions. I see some additional questions coming in. So for those of you who have put in some questions, don't worry. We will make sure that the speakers get your questions and make sure that you have your questions answered. And also feel welcome to continue to send questions in, and we'll make sure that they're answered. So thank you again for everyone that joined today. We hope that you all benefited and learned some useful information in this session. We will be administering a final survey in the next few days, so please take a moment to answer the questions on that. We would like to improve the work that we do. The recordings and the slides for all the presentations will be up on the website, and I will be letting everyone know when those are available. So again, we'll be sending out an email about that shortly. And please remember to register for the next session, which will take place on Monday, April 27th. So next week, we'll be taking a break, but we will be back again with another session on the 27th at 2 p.m. Eastern. That's a Monday. And for this session, we'll be joined by Elizabeth Arndorfer with the Reproductive Health Technologies Project, and also Martha Arguello with Physicians for Social Responsibility in Los Angeles, and Maya Nye with People Concerned with Chemical Safety. For this session, we'll be discussing the community grassroots perspective and local policy, particularly focusing on the grassroots advocacy perspective about community efforts in reducing and preventing exposure as it relates to oil and gas. So I think that's going to be a really interesting session. So I hope that you all will join us for that. So again, feel welcome to reach out to me should you have any questions. And we look forward to seeing you all at the next session. So thank you again to all the panelists and to Carolyn also for doing a great job moderating. So thank you, everyone.